Understand glycolysis. So glycolysis means lysis means breakdown, and glyco means glucose. So lysis of glucose that will give us usable energy. Now note that this whole process is irreversible and it happens in cytoplasm. The goal is to convert glucose into pyruvate, which are the usable form of glucose. So in short, the whole thing is that glucose is added with 2 phosphates plus 2 ADP plus 2 NAD that give us 2 molecules of pyruvate, 2 molecules of ATP, 2 molecules of NADH, and 2 hydrogen and 2 water. So that's the whole story. What happens that when we eat sugar, when we eat basically the glucose gets converted into glucose 6 phosphate. Now this conversion happens with two enzymes, or okay, the thing is, or either this enzyme can be used or this enzyme can be used. What can be used? Either glucokinase can be used or hexokinase can be used. Now glucokinase has really high km value and really low affinity. What does it mean? It means that it really doesn't love the glucose kinase. What does kinase do? Kinase is actually a phosphatase. It adds the phosphate group, okay? And hexokinase, it also a phosphate group. It also adds the phosphatase. So what's the story? The story is that it has low affinity for glucose 6 glucokinase. It means it only turns the glucose into glucose 6 kinase when it's an emergency. So it acts like a sensor. It's an emergency brake. So where do we need emergency brake? We need emergency brake where we need to have glucose levels monitored, right? So where we need to monitor glucose level, we need to monitor them in liver and pancreas, because both of them are big glucose unit. Liver makes insulin, pancreas makes insulin, and liver makes these enzymes that breaks down glucose. So we need to make sure that glucose is constantly checked through. So what happens is that this glucose cannot enter inside the liver without this enzyme called glucose kinase. So glucose first gets converted to glucose 6 phosphate, then it can enter. Another thing what hexokinase can do is that it has low km value and high affinity. What does high affinity means? High affinity means it loves the glucose, right? It makes sure that the glucose levels in your serum is constant, right? It keep converting this glucose level into glucose 6 phosphate. And when I'm saying it's a constant thing, it means it's doing constantly. It means that it's present in all cell. So that's the story. So glucose get converted into glucose 6 phosphate. Later glucose 6 phosphate is converted to fructose 6 phosphate. Now here is an enzyme as well, but I didn't need to put a name into it because it's not clinically really significant. Other thing is that glucose 6 phosphatase later converts into fructose 1,6 biphosphate, and it is converted with the help of phosphofructokinase 1. So there are two enzymes, phosphofructokinase 1, PFK1, and another enzyme is PFK2. So PFK1 converts this phosphose 6 phosphate into fructose 1,6 bisphosphate, right? So this is done how? Well, this enzyme PFK1, this enzyme is promoted by AMP, that is mono. What is ATP? ATP is molecule. It's an energy packet which has tri-phosphate molecules, okay? Tri, so it's an energy. But this AMP, it's mono, right? So it means AMP is less energy, right? And ATP is more energy. So if you already have enough energy, why do you even need to do glycolysis, right? You don't need to do glycolysis. You don't need to break down this. You don't need to break down glucose into energy packets because you already have ATP. So that's why ATP citrate negatively, inhibits this phosphofructose kinase 1, and AMP, the mono thing, the mono molecule which has less energy. Of course, we don't have energy, so we don't break down the glucose. So it promotes the phosphofructokinase 1. Other thing is that fructose 6 phosphate can be converted into fructose 2,6 biphosphate using PFK2. Now this PFK2 is making fructose 2,6 biphosphate, and the work of fructose 2,6 biphosphate is to promote PFK1. So again, what's the work? the work of fructose 2,6 biphosphate is to promote PFK1, right? Other thing is that PFK1 is positively promoted by insulin. Again, insulin is one of the anabolic hormone. 51 amino acid, 51 or 52 like that. Big protein molecule. Glucagon is the hormone that works when glucose is gone. So it works negatively. It inhibits PFK2, right? So once glucagon inhibits PFK2, fructose 2,6 biphosphate won't be formed. It won't be able to promote PFK1. So later PFK1 converts fructose 6 phosphate into fructose 1,6 biphosphate. Now note that whenever there is addition of one phosphate, we call it biphosphate, and then there's the addition. So 1 is the position, right? For example, here fructose 6 phosphate, what happens is that this 6 phosphate converts into biphosphate, and the position at which it happens is that at the number 2, and this happens at number 1. So this, I'm giving you a chemical structural meaning to it. Later fructose 1,6 biphosphates gets converted into phosphophenol pyruvate, and now the most important thing is that this phosphophenol pyruvate gets converted into pyruvate via an enzyme called pyruvate kinase. So there is a kinase. Now, if kinase is there, it means it's adding a phosphate group. There are a lot of enzymes named kinases. It means they cut the kinase, right? So kinase converts the pyruvate into this phosphophenol pyruvate into pyruvate, and again, just your common sense, pyruvate kinase is inhibited by ATP citrate, because ATP citrate it means that cell already has enough energy, right? So why do you need to break the glucose, right? So ATP citrate inhibits pyruvate kinase, and then it results as pyruvate, So the whole glycolysis can be summarized as glucose plus 2 phosphates plus 2 ADP, 2 NAD gives us 2 pyruvate, 2 ATP, 2 NADH, 2 hydrogen, 2 water. So the goal was to take glucose, convert into pyruvate. Rate limiting step is PFK1, and one of the thing is glucokinase is formed in liver and pancreas. Hexakinase is found in all cells. So this was glycolysis.